Hello, my name is Vicky Ia, and I will be talking to you about the investigation of a PET imaging probe to detect device-associated infections. Surgical implants such as dental implants, vascular stents, catheters, and orthopedic devices often result in infections that put a major burden on the healthcare system. Ideally, we would detect the infections early on rather than waiting for symptoms to arise. However, the typical detection methods in the clinic are invasive, lacking in sensitivity, and time-consuming. Therefore, the field has been developing novel imaging approaches to detecting pathogen-specific infections. Let's talk about glutamine. Glutamine is an amino acid that's important for bacterial processes such as survival, growth, and virulence. It mainly comes in two forms, L-glutamine and D-glutamine. You may compare these to a pair of shoes. They're exactly identical, but you can't wear your right shoe on your left foot. Likewise, humans can use L-glutamine, but they cannot use D-glutamine like bacteria can. In our study, we use PET imaging and a D-glutamine-based imaging probe to target and visualize bacterial infection in a rat model to evaluate its potential use in the clinic. Meanwhile, L-glutamine served as our control. To begin, we first wanted to establish a baseline for L-glutamine versus D-glutamine in bacteria. To do this, we chose the bacteria E. coli and Staphylococcus aureus, which represent more commonly found gram-negative and gram-positive bacteria associated with device infections. The probe was synthesized with a carbon-11 as the radiotracer, then incubated with cells for 30, 60, and 90 minutes. When the cells were collected and the radioactivity measured by gamma counter, we found a 10 to 12-fold higher uptake in the L-glutamine compared to D-glutamine. This was expected since L-glutamine is extensively used by both bacterial and mammalian cells. But there was also, interestingly, a greater increase in uptake rate in D-glutamine. Now, when we threw in some unlabeled amino acids to be actively metabolized, the uptake dropped by about 50% at a certain concentration of the unlabeled amino acid. And this suggests that there's an active mechanism involved for specific uptake of both the L and D-glutamine. We also showed that D-glutamine could be more broadly applied to different pathogens, and because of these differences, it could also be used to distinguish between the different pathogens. Next, we wanted to know if there is greater uptake of D-glutamine in bacteria compared to mammalian cells, as we predicted. Our results showed an 8-fold increase from 30 to 90 minutes of incubation, while the D-glutamine remained largely unchanged. When studied in mice, we found that L-glutamine uptake was higher than D-glutamines in most organs, with the kidney being the exception, and this indicates that D-glutamine ended up collecting in the kidney to be excreted after non-utilization by the other organs. Finally, we prepared a rat infection model using steel screws as an implant, and the PET images showed high sensitivity for the D-glutamine at the infected site, while L-glutamine did not. Histological analysis confirmed the presence of bacteria at the infected site, while the sterile site only showed inflammation. In summary, we were able to show in our proof-of-concept study that D5-carbon-11 glutamine, our tracer, is actively processed in both gram-positive and gram-negative bacteria, but not by mammalian hosts. This D-amino acid-based probe thus has clinical potential for PET imaging of device-associated infections by specifically targeting active bacterial processes.